continuing medical education and continuing education. For APA credit, there's an iStart ambassador towards the back of the room who's waving right now. And you can sign into those sheets, and you can visit the AIC Continuing Education website if you have any questions. So I'm so excited about this session that we put together today, talking about pointer imaging baseline results in relation to other studies and exploring generalizability from the perspective of the unique pointer sample. My name is Susan Landau. I'm PI of the Pointer Imaging Ancillary Study, and this is Prashanthi Murray, my co-chair, who is one of the MRI core leads. And she's going to give an introduction.
It's a pleasure to have all of you here. Good afternoon. Our goal for this session was to look at a population-based recruitment mechanism such as Pointer and compare it to other cohorts and try to understand and emphasize how a community-based recruitment is really important. And Pointer has done a great job with it. As you can see, it's 6 amazing speakers, all working with different cohorts, different variables of interest. And the question was, do our findings from clinic-based cohorts generalize to a population-based recruitment strategy?
So before we get started on the talks, since all the talks are going to be discussing pointer data, I wanted to say a few words about the pointer study design. I was thrilled and excited to witness the reporting of the successful pointer trial results yesterday. We have many of the pointer trial leads in the audience today, and just as a quick overview, so the JAMA paper reporting the successful results came out yesterday. There are other papers that cover the study design and the baseline characteristics of the pointer cohort. This was a population of about 2,000 older adult participants who were recruited from the community via grassroots outreach and targeted mass mailing. 5 US sites. They were assigned to self-guided and structured intervention groups, and they had cognitive testing at baseline and throughout the course of the 2-year study. I won't go through the inclusion criteria, but you can see this risk-based criteria on the screen here. And I think all of these unique aspects of how pointer participants were both recruited and also enrolled with these risk-based criteria kind of plays into this question of generalizability that we're going to talk about today. So the imaging sub-study is one of 4 ancillary studies, and you'll be hearing about this, this data in the coming months as we look at our intervention effects. We're incredibly excited about it. And so for today, we're going to be focusing on the baseline characteristics of this data set. There is some ancillary study data that's going to be reported in this session, and of course, other data, as I said, to come. So in the imaging study, we enrolled about 50% of the total main trial participant group. We had amyloid and tau PET at baseline and at 24 months, and had MRI scanning to look at brain structure and a variety of cerebrovascular measurements at baseline, 12 months, and 24 months. And you can see some of the key characteristics of the sample at the bottom there, and you'll hear more about this in the talks today. So our first speaker is Tessa Harrison from UC Berkeley, and the title of her talk is White Matter Hyperintensities and Alzheimer's Disease Pathology in Heterogeneous Cohorts.
Thank you. I'm going to take that introduction just a little bit further and say that we care a lot about generalizability in AD biomarker research. And concern really arises because we know that every cohort and every study is the result of that particular study's recruitment approach, the sites from which they recruit, and will reach different groups of participants. So today, I'm going to talk about 4 different studies. The one probably best well known to you all is ADNI. This is a clinical trial ready cohort of normals and AD patients with geographic diversity, but low cardiovascular disease due to exclusion criteria, and low ethno-racial diversity, especially in the first few phases of ADNI. Pointer imaging, which you just heard about and you'll hear about in all the talks today, this cohort is comprised of individuals who were enrolled in this multi-domain lifestyle intervention study, has geographic and ethno-racial diversity, higher cardiovascular disease burden, and participants were required to have poor lifestyle metrics. The SCAN cohort is comprised of individuals from U.S. Alzheimer's disease research centers. They are normals and patients, both AD and non-AD dementias. There's geographic diversity, some ethno-racial diversity, and significant clinical heterogeneity. And finally, HABS-HD. This is a study of aging in AD. There's no geographic diversity as it's a single-site study, but equal-sized ethno-racial groups. And so one approach to sort of addressing generalizability is to pool data, harmonize and pool data across samples to increase the heterogeneity of your pooled sample and also the statistical power to enhance generalizability and reproducibility. The other way you can think about this is you can compare cohorts to understand the impact of specific characteristics on biomarker relationships. And I'm going to do a little bit of both in this presentation today. I'm going to focus on white matter hyperintensities. They are generally associated with ischemic changes in small vessel disease and have been used as a biomarker of cerebrovascular disease. But increasingly, it's understood that the etiology of white matter hyperintensities is very complex and may also be related to AD and the associated neurodegeneration. So in this project, we aim to explore associations between white matter hyperintensity volume and AD pathology first, on the one hand, and cardiovascular risk and evidence of cerebrovascular disease, specifically cerebral microbleeds, on the other hand. And further, we want to understand if these associations vary by the four cohorts I introduced in the last slide. We calculated white matter hyperintensities using a pipeline developed by the DeCarli lab at UC Davis. They used this pipeline to generate white matter hyperintensity data for ADNI, pointer imaging, and SCAN. So we were able to validate our Berkeley run of this software against those three data sets, but we wanted to make sure that all the data you'll see here today were generated at UC Berkeley for sort of maximum comparability across all four cohorts. This is the sample I'll be focusing on. So I'm focusing just on cognitively unimpaired individuals. We really care a lot about what's happening in this preclinical part of the phase of the Alzheimer's disease spectrum. And I'll just focus on a couple things. In our pooled sample, we have a mean age of 69, but you'll see that the mean age varies across the four cohorts in a way that affects our results. In the pooled sample, 59% of participants are non-Hispanic white, but this varies considerably across the four cohorts. And also, I just want to point out this centilloids row and note that the ADNI cohort has the highest mean centilloids because this is going to be pertinent to some of the results I'll show you in a couple slides. White matter hyperintensity volume increases with age. This is a robust effect that's been shown many times before. It's also higher in females. Again, this is an established effect. So in order for us to do cohort comparisons and feel really good, we're comparing fairly. We're going to adjust our white matter hyperintensity volumes not only by intracranial volume, but also by age and sex. So most of the plots I'll show for the rest of the presentation have this age and sex adjustment for the white matter hyperintensity volumes. If we just look at white matter hyperintensity volume by the four cohorts, the only pairwise difference that is significant here is that the HABS-HD cohort had higher white matter hyperintensity volumes than the other three cohorts. So we're going to start off by looking at, in terms of our biomarker associations, looking at global amyloid associations with white matter hyperintensities. And you can see that there is an association, but it's a small effect. And if we look at this across the different cohorts, you see that this effect is strongest in ADNI. This might be related to the fact that ADNI was really recruited as a AD-specific sample and has the highest mean centilloids. When we look at the associations between white matter hyperintensities and tau, specifically in the entorhinal cortex, because again, these are all unimpaired individuals, and so we're focused in this early region. We see there's no association in the pooled sample, but in each cohort individually, we see there's an association only in ADNI, so a similar story from the last slide. Now, switching gears a little bit, we're going to look at the relationship between white matter hyperintensities and Framingham risk score. Framingham risk score is a cardiovascular disease risk score that captures the 10-year risk of a cardiovascular disease event. It's comprised of age, sex, systolic blood pressure, BMI, diabetes status, smoking status, and whether or not you're taking a blood pressure lowering medication. And you can see there is an association between FRS and white matter hyperintensity volume in the pooled sample. And this effect is mostly consistent across our cohorts, despite the known differences in cardiovascular disease burden in these cohorts. But whenever we see an association with FRS, we think to ourselves, okay, well, which of the parts of this, components of this association is driving the effect? And so you'll see here I didn't adjust for age and sex on the y-axis here because we know that age and sex is part of the FRS and driving this effect in part. But when we look at the other parts of the FRS score, the one that showed a relationship to white matter hyperintensities was systolic blood pressure. So we see in the pooled sample, this is a very small effect size, so we wouldn't really expect this to be significant in a smaller sample. And indeed, when we look across all of the 4 cohorts, it's only significant. It's remarkably consistent, but it's only significant in our 2 largest samples. Now we're going to look at how white matter hyperintensity volume is associated with a marker of cerebral vascular disease in the brain, namely cerebral microbleeds. So the presence of 1 or more cerebral microbleeds is a strong predictor of higher white matter hyperintensity volumes. We double and triple check this in this particular subset of the scan data set. There's only 1 individual who had a microbleed, so we can't do group statistics on that cohort alone, but you can see it's consistent across the other 3 cohorts. And we know cerebral microbleeds indicate that there's been a small hemorrhage, so related to small vessel disease or CAA, both of which are suspected etiologies of white matter hyperintensities. So we were also interested in looking at white matter hyperintensity status. So we defined white matter hyperintensity positivity as the top tertile of our age and sex adjusted white matter hyperintensity volumes across samples, so in our pooled data set. And this is just a little bit of a review to show that we see the same pattern of effects using this dichotomous approach compared to the continuous approach. So again, a subtle effect of global amyloid, strongest in ADNI, no relationship with enteronal tau except in ADNI, and an association with FRS and cerebral microbleeds. But this is all teeing up that we want to put all of these measures into the same model. So this is the results of a logistic regression predicting white matter hyperintensity status. And you can see that even though the overall effect association between global amyloid and white matter hyperintensity volumes is small, it independently predicts white matter hyperintensity positivity along with systolic blood pressure in this model. And when we look at the 4 cohorts of the same model individually, you can see there's quite a bit of variability as to whether or not these predictors are significant. So I think having these 2 plots side by side is really demonstrates the power of harmonizing and pooling data to try to get at sort of the heart of these associations, even especially when the effect sizes are small. This is now a second logistic regression where we've added that dichotomous cerebral microbleed variable, and you can see that's a very strong effect. But actually, amyloid and systolic blood pressure remain significant predictors in this model as well. And this is what this model looks like when you look at the cohorts individually. Again, a good amount of variability there. So to wrap up, in our large, over 2,500 individuals, sample of cognitively unimpaired older adults, we found that global white matter hyperintensity volume was weakly associated with amyloid, and this effect was strongest in ADNI and was not associated with tau except in ADNI. We found that the Framingham risk score was related to global white matter hyperintensity volume, both in our pooled sample and pretty consistently across cohorts, and this was really driven by age, sex, and blood pressure, systolic blood pressure. The presence of at least 1 cerebral microbleed was a strong predictor of higher white matter hyperintensity volume, and this effect was also consistent across studies despite their very different characteristics, especially cardiovascular disease burden-wise. And despite the general small effect size of the association with amyloid, our logistic regression still showed a significant effect for global amyloid predicting white matter hyperintensity status alongside the vascular measures. So in the future, we have a lot more work to do with this project, and one of the things that's top of my list is looking at white matter hyperintensities regionally. There's been some work in the field suggesting that anterior white matter hyperintensities are more related to vascular disease and vascular burden, and posterior white matter hyperintensities more related to AD, specifically amyloid. We also want to integrate cerebral microbead location into this because we know that deep subcortical CMBs are more likely to be related to hypertension, and cortical CMBs are more likely to be related to CAA. So we want to understand, are there different associations with white matter hyperintensities in these two categories, and how does amyloid fit into that picture? And finally, we want to harmonize cognitive data across studies so that we can understand white matter hyperintensities, contribution to cognitive performance, which has been looked at in many studies, but also especially alongside the other measures we have here in this cohort. And with that, I will thank the ADNI SCAN, US Pointer, and HABS HD participants, sponsors, and staff, and all of my colleagues at the JAGUS Lab at UC Berkeley. Thank you.
Our next speaker is Pauline Maillard, and her talk is Risk Factors and Clinical Relevance of MRI Vascular Markers in the US Pointer Study, a cross-cohort comparison with the Framingham Heart Study.
Hi everyone, I'm very pleased to present our work on vascular risk factor, MRI marker of vascular injury and cognition in the Pointer Study. This project is part of a cross-cohort comparison with the Framingham Heart Study, and it examines the generalizability of biomarker, imaging biomarker relationship across different populations. So cerebrovascular disease is a major contributor to cognitive decline and dementia. MRI markers search white matter hyperintensities, free water, fractional anisotropy, and perivascular spaces, capture early vascular-related brain change. And the US Pointer Study is a lifestyle intervention trial of cognitively normal adult participants at risk for dementia. And our key question was whether at the beginning of the study, before the intervention occurred, the relationship between vascular risk factor, these MRI markers, and cognition observed in the Pointer Study can be generalized to other populations. So our goal was to examine the association of 4 key MRI marker of vascular injury, including white matter hyperintensities, free water, fractional anisotropy, and perivascular space, with both vascular risk factor and cognition. And we compared results across 2 cohorts, including the US Pointer Study. As I just mentioned, this is a lifestyle intervention study including older individuals at risk to convert to dementia, and the Framingham Heart Study, the offspring cohort, which is a long-standing population-based observational cohort with decades of follow-up. So we focused on 4 imaging measures of vascular injury, including free water. Free water is derived from diffusion tensor imaging, and it reflects the amount of water molecule in the white matter tissue that are not constrained by their local environment. Fractional anisotropy, which is also derived from diffusion tensor imaging, and it reflects the integrity of the myelin of the white matter tracts. White matter hyperintensity, which is a more renowned marker associated with small vessel disease and likely represent the most advanced stage of white matter degeneration. And perivascular space, which is a fluid-filled compartment around the blood vessel. So higher level of free water, higher level of white matter hyperintensities, or higher level of perivascular space, or lower level of fractional anisotropy, each indicate a higher increased amount of vascular injury in the brain. So free water and fractional anisotropy were generated using the same method in both Pointer and Framingham. We use free water kits that we developed for the MarkVCID consortium. As Tessa mentioned, white matter hyperintensities have also been generated using the same method, the UCD white matter hyperintensities segmentation toolbox. Only perivascular space was measured differently between Framingham and Pointer. In Pointer, we used volumetric measure using a method developed by Lisa Silberg from Oregon University. And for Framingham, perivascular space consisted of score from 0 to 3 according to the burden of PVS. And that work was done by Dr. Rafael Romero from Boston University and collaborator from, of the Framingham Earth study. So all these biomarkers provide complementary views in terms of chronology or biomechanism of vascular injury burden in the brain. So this slide summarizes two study population, and while Pointer and Framingham both include older participants, impaired cognitively, they differ in terms of ethnic, racial composition, in terms of inclusion criteria, but also in terms of study design. The US Pointer targeted participants who are at risk to convert to dementia, and it includes a very high proportion of underrepresented group. The Framingham study, in contrast, it's an observational study, and the participants are mostly non-Hispanic white participants. But this contrast makes actually these two studies ideal for examining generalizability. To assess cognition, we focus on executive function, which we know is a cognitive domain very sensitive to vascular injury in the brain. And executive function composite score were actually available for both studies. And you can see an overview of how these scores were computed. So it's a bit different. And while the method and the specific test batteries differ between these two studies, it's important to understand that the two approaches capture the same underlying cognitive construct. So we use linear and logistic regression to investigate the effect of vascular risk factor on the MRI measure and of the MRI measure on cognition, adjusting by demographics and relevant covariate. For the cognition model, we included education as well as the gray matter volume to take into account the effect of neurodegeneration on cognition. So the next few slides, I'm just going to describe the results of the association between vascular risk factor and all 4 MRI markers, including free water, FA, white matter hyperintensities, and PVS in the US Pointer study and in the Framingham Earth study. So first, looking at hypertension, we found that hypertensive participants had a higher level of free water, of PVS, and a lower level of fractional anisotropy in both US Pointer and the Framingham Earth study. We also found that individuals with diabetes had higher level of free water in the US Pointer study and in Framingham Earth study, but was not associated with any of the other biomarkers in both studies. We did not find significant effect of smoking status on the 4 biomarkers, neither in US Pointer or the Framingham Earth study. We found a very strong effect of age, increasing age being associated with increased free water, increased white matter hyperintensities, increased PVS, decreased fractional anisotropy. We observed the same association in US Pointer and Framingham. Regarding sex, we found different effects of sex according to the biomarker and the study. Now, turning to the association between the imaging marker and executive function, we found a significant association between higher level of free water and lower level of cognition. You can see the association in the graphic here between free water and executive function for US Pointer and Framingham Earth study independently. We also found an association between increased white matter integrity as reflected by increased fractional anisotropy with executive function. Again, you have the association in this figure right here. We did not find association between white matter hyperintensities or PVS with executive function in either US pointer or Framingham. So to summarize, despite the very different cohort characteristics between pointer and Framingham, we found consistent association between all 4 MRI markers of vascular injury with vascular risk factor and with cognition, which supports the generalizability of findings from the pointer study. In terms of clinical relevance, we also found that free water and FA seems to emerge as more sensitive marker for cognition as compared to white matter hyperintensities and PVS. And all this work also highlight the value of cross-cohort comparison for validating imaging biomarker. And I would also like to thank all the participants from the US Pointer and Framingham Earth study, as well as all the staff and collaborators from Framingham and Pointer study, and all my team at UC Davis. Thank you for your attention.
The next speaker is Pei-Wei Liu. Does cohort heterogeneity affect relationships between AD biomarkers and cognition?
Okay, thank you for the introductions and having me in this session today. I have nothing to disclose. So in the current study, I focus on the pointer dataset because this dataset is a more representative sample and is community recorded study that emphasizes the geographic diversity, inclusions of underrepresented populations, and enrichment for the cardio risk profiles. One interesting finding in our lab showed that compared to the matched pointer dataset, the more homogeneous ANI sample shows more stronger associations between inter-high tau and global cognition, especially among the amyloid positive individuals. So we would like to know why they have this kind of interesting finding. So firstly, we would like to know whether this finding could replicate or extend to the more specific cognitive domains, such as verbal memory and executive function. Second, whether the greater vascular risk in US pointer can contribute to this cohort difference in the tau and the cognition relationships. Third, we would like to know whether the brain atrophy could contribute to this finding as well. So this study is focused on the baseline data in the pointer, and we also extract the data in the ADNI, which the data collection time is most close to the tau collection time in the pointer. So we did the cohort matching first because we would like to make the two cohorts comparable. For example, we would like to make sure they have the similar clinical cognition conditions, and we would like to remove any the potential confounding effects. So firstly, we excluded the participants without the key imaging biomarkers in both cohorts, and we also excluded the participants in the ADNI only with the moderate to severe cognitive impairment because there's no significant cognitive impairment in the pointer dataset. So after that, we conducted the propensity score matching based on the age, sex, ApoE4 status, and the CDR sum of boxes. The final sample size in pointer is 775, and in ANI is 405. So this table shows the basic information of the descriptive stats in the matched cohort. So we can see the matching variables did not differ significantly between the two matched cohorts, except the age, but the age difference is very small, it's less than 1 year, so that means over cohort matching works. And for the cognition, you can see that the pointer shows the worst performance in the majority of the cognitive tests. But CDR sum boxes, there's no cohort difference because they use it for the cohort matching, so that makes sense. And for the cardiovascular risk, the pointer shows the higher risk profiles in terms of the BMI and the smokers. But there's no cohort difference in the systolic blood pressure or diabetes. And for the biomarkers, means there's no cohort difference in the amyloid positivity percentage and high no tau, white matter hyperintensity, hippocampal volume. But the pointer shows the last tau in the metatemporal regions here. One thing I want to mention here that there's the different tracers used in the pointer and adenine. For pointer, we use MK6240 and adenine use the FTP. So we just calculate, we need to standardize the MK and FTP by the z-score based on the mean and standard deviation from the reference group. The reference group with the participant is cognitive impairment, amyloid negative, and the age is 6 to 7 years old. Okay, so in response to the M1, whether this finding, the cohort difference in the tau and the cognition relationship could extend to the specific cognitive domains. So we fit a couple linear models. So you can see among amyloid negative individuals, there's no associations between anti-hydro tau and 3 cognitions in the both cohorts. But interesting that among the amyloid positive individuals, there's a significant tau by cohort interaction. So which means compared with the matched pointer, the adenine individuals shows the significant negative associations here. And we also look at the metatemporal regions. The pattern is pretty similar, except the tau by cohort interactions among the amyloid positive individuals when we look at the verbal memory. So in summary here, it seems over previous finding could extend to the specific cognitive domains. So over remaining analysis, we will only focus on the amyloid positive individuals because we only, our research aim is to examine the reason why there is a cohort difference. Okay, for the result 2, we are interested in whether the vascular biomarkers could contribute to this. So we fit a couple linear model as well. First of 4 graph, I show the non-neural imaging-based vascular biomarkers. So you can see the more systolic blood pressure is associated with the worst global cognition in pointer. And for BMI, the higher BMI was associated with better verbal memory. So this result is a bit counterintuitive, but the previous study showed that the BMI-related associations in older adults is pretty complex. It may reflect the reserve in older adults. For example, the higher BMI may reflect the lesser possibilities of undernutrition or the greater mass, right? Good muscle mass. So we need to look into more here. And for smoking, there's the 3 cognition did not differ between the 2 smoking groups. For the diabetes, executive function performs better in the pointer without the diabetes compared to the counterparts. And we also look at the neuroimaging-based vascular biomarkers. So you can say here, the greater white matter hyperintensity volume was associated with the worst executive function. And for the CMB, the verbal memory performs better in pointer dataset only for the people without the CMB compared to the CMB. So 3 things I will mention here. The associations here is pretty weak. It cannot survive after the model comparison correction. And there's no cohort by vascular biomarkers interaction. So even you see some significant result only in the pointer. And finally, the total variance explained by these vascular biomarkers was pretty small and is below than 6%. So in total, the vascular biomarkers could not explain too much variance in the cognition and cannot explain too much about the cohort difference between the tau and cognition relationships. And third, we would like to know whether the cortical thickness could contribute to this as well. So we conducted the correlations between the regional cortical thickness and 3 cognition with interhydral tau as one of the covariates. And the contrast results show that compared with the pointer, the cortical thickness in adenine in more than 25% regions showed greater positive associations with the cognition, especially on the global cognition and executive function. So it seems the cortical thickness, that is the tau independent role to explain the cognition. So in conclusion, the previous finding the stronger and the widespread tau cognition associations in any then pointer could extend to the specific cognitive domains, especially among amyloid positive individuals. And some vascular biomarkers were weakly associated with cognition US pointer amyloid positive individuals. So the vascular biomarkers are not a major factor in explaining cohort difference in tau and cognition relationships. And cortical thickness showed stronger and broader associations with cognition, especially on the global cognition and executive cognition in any amyloid positive individuals, even after adjusting for the interhynal tau. So there's a tau independent role of cortical thickness in explaining the cohort, but this does not explain the cohort difference in the tau and cognition relationship. So we need to look into this question more with more other impacting factors here. And this study also indicates that the cohort recruitment differences impact the generalizability of AD biomarker research. So I would like to say thank you to the lab members in Jack's lab and the entire amazing Pointer team and AN team and the AIC fellowship. Thank you.
Thank you, Pewe. Our next speaker is Carmen Calzeriu. She's from Barcelona Global Health Institute, Barcelona, Spain, and she's going to show us on an international perspective on social demographic associations with AD biomarkers.
Good afternoon, everyone, and thank you for the introduction. Socioeconomic deprivation has been shown to be a risk factor for a wide spectrum of poor health in adults, including in AD and dementia. Neighborhood deprivation has been linked to specific demographics, such that people living in highly deprived areas are usually younger, more often females, and less often college-educated or white. Living in a deprived area has been associated with slightly faster cognitive decline, higher odds of AD neuropathology, and in a very few studies with lower hippocampal and total brain volume. However, results often vary across cohorts. And this may be due to differential demographical composition of these cohorts, such as in race and ethnicity or geographical location, which leads us to our research question of today of how generalizable are the associations between neighborhood deprivation and AD biomarkers across cohorts. Our aim here was to assess the consistency of these associations among 3 cohorts with different recruitment strategies. The cohorts that we were comparing are ADNI, US Pointer, and UBrain, which is a cohort from Spain, which is leveraging retrospective data from 2008 to 2023, acquired in 2 of the main hospitals in Barcelona, in Hospital Clinic and Hospital Sant Pau. Because participants in UBrain are coming from a clinical setting, we decided to set a threshold of a minimental higher than 24, so we can filter out MCIs and participants with dementia, and we ended up with a cohort of 901 participants and different Ns for each biomarker. Similarly, in ADNI, we set a threshold of a CDR lower than 0.5, and we ended up with a cohort of 300 participants. In US Pointer, we used the full cohort for cognition and the full cohort for imaging and amyloid. Area deprivation index is a composite measure used to quantify socioeconomic disadvantage at the neighborhood level. And we use validated indices for each country. So for US Pointer and ADNI, we use area deprivation index from the US, which is composed of 17 indicators on different subscales, such as poverty, housing, and employment, and is divided in 3 levels of low, medium, and high deprivation by pre-established cutoffs. In UBrain, we use the deprivation index validated in Spain, composed of 6 similar indicators, and divided into 2 levels of low and high deprivation by the medium, because here, for this index, we don't have pre-established cutoffs. Our outcomes of interest were cognition, assessed through the preclinical Alzheimer cognitive composite in all 3 cohorts, amyloid status derived from CSF in UBrain and derived from PET imaging in ADNI and US Pointer, and gray matter volume in AD vulnerable regions, assessed with T1 weighted images averaged into a meta-ROI composite. For statistical models, we use linear and logistic regression models with ADI as a predictor, and we adjusted for age, sex, ApoE epsilon-4 status, and intracranial volume in the case of the meta-ROI. For the significant associations, we then added educational level as a covariate, and if the results were still significant, we then stratified by 3 educational levels. This is a table comparing demographics and clinical characteristics between the 3 cohorts. And we found that there are older participants in ATNI and lower CDR scores, higher percentage of females in US pointer, and lower education and mini-mental scores in new brain, even though we use the mini-mental as a threshold, which might be because participants are coming from a clinical setting. When comparing by biomarkers, we found lower PAC5 scores in US pointer, higher percentage of amyloid positive participants, and lower meta-ROI volume in new brain. This is a table comparing the demographics by ADI levels within each cohort. And first, I want to point out that in US pointer, there were only 10% of participants coming from highly deprived areas, and in ATNI, only 13%. Then we observed that in US pointer, there are more females coming from highly deprived areas, and no difference in APOE4 status by ADI level in any of the cohorts. In terms of race and ethnicity, I want to point out that in U-Brain there was no data available for this, and in US pointer and ATNI, we saw that in general there are more white participants in all ADI levels. However, there were more African Americans residing in highly deprived areas. We observed lower education in highly deprived areas as in all cohorts, as we would expect, and no differences by ADI levels in amyloid status or diagnosis. Now I'm going to show you the results of the associations of ADI with AD-related outcomes. First, for cognition, we found significant associations in US pointer, such that participants coming from moderate and highly deprived areas showed poorer cognition compared to participants coming from low deprived areas, while no associations were found in ATNI and new brain. However, there was a trend in new brain going in the same direction as in US pointer. When we adjusted by education, the associations in US pointer remained significant, while the trend in new brain became non-significant. Then, when we stratified by educational levels, first I want to point out that in US pointer there were no participants with less than 6 years of education. And then second, we found that the results are driven by highly educated individuals. Interestingly, in new brain, we found a reverse trend in highly educated participants who showed better cognition than any other group, independently of deprivation, which might suggest that in Spain, education can act as a buffer against the negative effect of deprivation. Then for amyloid status, we didn't see any significant associations in any of the cohorts, suggesting that participants had the same odds of being amyloid positive independently of deprivation. And lastly, for gray matter volume, we found significant associations in US pointer again, with participants residing in moderately and highly deprived areas showing lower gray matter volume compared to participants coming from low deprived areas. In ATNI and new brain, there were no significant associations, and when we adjusted by education, the association in the moderate ADI level became non-significant. Then, when we stratified by educational level, we found that again, the results are driven by highly educated participants. In conclusion, the associations between area deprivation index and both cognitive performance and brain volume were not consistent across cohorts. However, when looking at specific cognitive domains, we observed ADI cognition associations in new brain, as you could see today in my poster with the number 835. Second, the US pointer cohort did show significant associations between ADI cognition and brain volume, while only a trend in cognition was observed in new brain. This could be due to higher representation of race and ethnic minoritized group in the US pointer, as we have seen in our other works that race and ethnicity really does matter. And the limitation to consider is that there are no established cutoffs for ADI levels in Spain, limiting the significance of these deprivation levels. Third, the trend observed in new brain was inversed in highly educated participants, while the effects of ADI remain significant independently of education in US pointer. Pending further analysis, this may indicate country-level differences in access to education. Overall, differences in sample characteristics, including socioeconomic status, racial and ethnic composition, and recruitment strategies, may help explain the discrepancies across cohorts. Future studies examining health disparities and related biomarkers should prioritize diverse and representative cohorts to allow for more generalizable and equitable findings. I would like to thank my team from Institute of Global Health Barcelona, our collaborators from Hospital Clinic and Hospital San Pau, the US Pointer and US Pointer Imaging study teams, and all of you for listening. Thank you.
Thank you, Carmen. Then our next speaker is Talia Robinson. She's from MGH, and she's going to talk about these findings in non-Hispanic Black and non-Hispanic white participants.
Thank you for that introduction. I'm happy to present this work evaluating AD biomarkers in cognition in non-Hispanic Black and white participants in the ADNI and US pointer imaging cohorts, as well as some new additional analyses looking at a combined multi-cohort study that also includes A4 participants. So the motivation for this work is based on the low representation of ethno-racially diverse populations in many AD neuroimaging studies and how this may influence the generalizability of biomarker associations to these groups. In work that has been done, there is a trend of finding lower amyloid burden among non-Hispanic Black participants relative to non-Hispanic white participants, and this difference is found despite Black participants' higher risk of cognitive decline and dementia. And so this figure on the right is showing work taken from the A4 study in the orange bars depicting lower amyloid burden among Black E4 carriers relative to white E4 carriers, again in the A4 study. However, it remains unknown if this trend is present across other cohorts. It's also unclear if the biomarker associations that we see in primarily racially homogeneous cohorts generalize to other groups. And considering that Black participants or Black and African Americans in general tend to have higher burden of CV risk and cardiovascular disease, it's unclear if this influences biomarkers differently across ethno-racial groups. So we wanted to approach these questions in two ways. So in our primary analyses that were initially submitted for this symposium, we were looking at independent cohort analyses within the ADNI and US pointer imaging studies to characterize the cognitively unimpaired Black and white participants in these studies and evaluate their AD biomarker, CV risk, and cognition relationships, and assess if there is consistency in the relationship we see in these factors across ethno-racial groups and across cohorts. And more recently, we've conducted some combined multi-cohort analyses where we've combined data from the ADNI, US pointer, and A4 studies to evaluate, again, the Black and white participants and further explore these risk, biomarker, and cognition associations in hopefully a larger and more representative sample of Black participants. So this approach in these primary analyses, again, looked at the cognitively unimpaired Black and white participants in ADNI and US pointer. We took their baseline amyloid data, which was reported in scintilloids, entorhinal tau data, which was quantified differently in each cohort, but reported separately, as you'll see, and the preclinical Alzheimer cognitive composite. So for the US pointer study, this includes everyone's baseline data, and for the ADNI study, this includes the earliest tau scan and corresponding amyloid PET and cognitive data. And our statistical approach included within cohort and within ethno-racial group analyses. So for example, in the US pointer study, in the Black participants, we looked at the associations of CV risk and biomarkers and cognition, as well as biomarkers and cognition. And then we also looked at within cohort interaction models to assess these associations and if there are any differences by ethno-racial group. And I do think it's important when evaluating ethno-racial differences to look at both within group and interaction models to really understand what's happening in these associations within a group and not just only relative to another target group. So these analyses are completed in both cohorts. When we look at these samples, we see some consistencies in that the Black participants in both cohorts tend to be younger than the white participants, tend to have a lower mean years of education, and are disproportionately female. They also tend to have lower mean PAC scores. And when we look at the cohorts, again, we see that there are group differences or ethno-racial group differences in amyloid and entorhinal tau in pointer. So specifically in the pointer cohort, we see that Black participants have lower amyloid and lower entorhinal tau. However, in ADNI, these group differences do not survive corrections for age and sex. And when we look at within group, within cohort biomarker risk and cognition associations, we actually see pretty similar associations in both ethno-racial groups across both cohorts. So in the pointer study, we see an expected positive association between amyloid and tau in both groups. We also see the expected negative association with higher entorhinal tau associated with worse PAC performance in both ethno-racial groups. Interestingly, we see a negative association with BMI and entorhinal tau in the white participants, and we see no associations in CV risk and amyloid or CV risk and PAC. And in the ADNI study, we see very similar patterns of association. Now, however, this time we see that significant negative association between BMI and entorhinal tau in the black participants. When we look at interaction models of these different associations, we do see some differences in the strength of the association between CV risk and tau across groups and cohort, but no differences in the associations of biomarkers and cognition. So again, in ADNI, we see this significant negative association between BMI and entorhinal tau is different or stronger in the black participants relative to the white. However, in Pointer, we see no differences in the association of BMI and tau across ethno-racial group. When we look again at that positive association between amyloid and tau, we see no differences in the strength of this association across ethno-racial groups in either cohort. And when we look, importantly, at the association between entorhinal tau and cognition, this negative association, again, is not different by ethno-racial group in either cohort. And so now I'll quickly talk about these combined multi-cohort analyses. So again, the motivation for this is, as was mentioned, each of these cohorts may have distinct characteristics based on the particular study goals, whether that be higher burden of CV risk, higher burden of amyloid, ethno-racial diversity. And so we hope that by combining the data from these three cohorts, US Pointer, Imaging, ADNI, and A4, we might hopefully develop a larger sample of Black participants with hopefully better representativeness, at least along these specific dimensions. And so we combined this cognitive, amyloid, and CV risk data in these three cohorts to obtain a larger combined sample of Black participants. And we used, just to note that tau data was limited in these Black participants. However, that remains a future goal with the advent of more gold standard harmonization techniques for different tau tracers. But after combining these cohorts to obtain this combined match sample of Black participants, we used propensity score matching to obtain an age, education, and sex match sample of non-Hispanic white participants. And again, we wanted to characterize these new match groups on their amyloid burden, CV risk, and cognitive performance, and evaluate associations in these factors and any potential differences in these associations by ethno-racial group. So the pie chart you see demonstrates the proportion of each cohort that's represented in this new combined match sample, again reflecting the availability of the data in the Black participants from each of these cohorts. And now when we look at this new combined match sample, we see that the groups are consistent regarding age, education, and sex. I will point out that based on the proportion of females in the Black cohort, we see that this new match sample is now disproportionately female. And again, we see that the Black participants tend to have higher mean BMI and higher mean diastolic blood pressure, although this diastolic blood pressure difference is statistically significant but likely not clinically relevant. And we also see the Black participants again have lower amyloid, but interestingly, the groups do not differ in terms of proportion of amyloid positivity or in E4 carrier status. The group differences that we see in amyloid and PAC, so the lower amyloid burden and lower PAC performance we see in the Black participants remains even after adjusting for demographics and these CV risk factors. And when we look at the associations between CV risk and amyloid, very quickly, there's no association overall between any of our CV risk factors and amyloid in these data. There's also no differences in within-group analyses or interaction models in this association across ethno-racial groups. But when we look at CV risk factors and cognition, we see that overall there is a positive association between BMI and systolic blood pressure, or negative association, between BMI, systolic blood pressure, and PAC performance. And when we look at within-group models and interaction models, we see no difference in the strength of these associations across ethno-racial group. And finally, there was overall a non-significant association between amyloid and cognition in this combined match sample. And again, in within-group models and interaction models, we see no difference in the strength of these associations by ethno-racial group. So in conclusion, we can observe that there are differences in ethno-racial group characteristics across cohorts. So the non-Hispanic Black participants tend to be younger, have a higher proportion of women, they have variably higher levels of CV risk factors, and lower amyloid burden rather consistently. However, importantly, these differences do not appear to influence the associations that we see between biomarkers and cognitive outcomes, at least in these cross-sectional analyses. So for future directions to expand upon this work, we hope to include other indicators of CV risk that might be better predictors or more relevant predictors of CV risk. We also hope to include other biomarkers, tau as I mentioned, and possibly other direct indicators of cerebrovascular injury. We also, of course, would like to include longitudinal cognitive outcomes to understand if these baseline differences are relevant for change over time or differences in rates of change over time, perhaps. And also, we would like to include further cohorts to have greater opportunities to have an even larger, perhaps more representative sample with other opportunities to evaluate important influences on cognition like psychosocial factors or social determinants of health. So I would like to thank my mentors and co-authors on this project, as well as the participants and study teams of ADNI, A4, US Pointer, and thank you all for your time.
Thank you, Talia. Our final speaker is Dr. Joe Weiner. He's going to talk about heterogeneity and association between short sleep duration and amyloid PET.
Thank you. Okay, so maybe focusing on sleep. So you're probably familiar with this idea that there's a link between sleep and Alzheimer's disease. One of the first landmark findings that linked sleep with AD biomarkers in humans came from the Baltimore longitudinal study of aging, which showed that cognitively unimpaired older adults who reported getting less hours of sleep had more amyloid PET burden, higher levels of amyloid in their brain. And when a few years ago when the A4 screening data were released, I was really excited to see that they actually asked everyone who was screened for the study how many hours of sleep they got. So we were able to look at this data compared to more than 4,000 amyloid PET scans, and we actually saw the same pattern, which was linear association with shorter sleep durations predicting higher levels of amyloid PET. And so this idea of a link between sleep duration and amyloid supported a hypothesis that actually came from mouse model studies, where APP mice who are bred to accumulate amyloid actually get less sleep over time as the amyloid aggregates, and they spend more time awake. And of course, there's now a growing field continuing this work of understanding this relationship between sleep and AD pathology, which probably involves a very complex interplay between overproduction of waste products in the brain, clearance of those waste products, and also changes in sleep physiology as pathology aggregates in circuitry that's important for sleep-wake regulation. But I'm not going to focus on any of those ideas today because in the A4 screening data set, amyloid was not the only correlate of sleep duration. We also found that female sex and higher education were both associated with longer sleep duration. And we also found that individuals from underrepresented groups slept less than the non-Hispanic white individuals. So while people who were non-Hispanic white averaged about 6.5 hours of sleep, we found that Hispanic Latino group slept about 15 minutes less. The non-Hispanic Asian group slept an average of about 30 minutes less, and the non-Hispanic Black group slept about 40 minutes less. And so we reported this in the paper, and I thought it was kind of an interesting finding in the midst of these amyloid and cognition relationships, but it has kind of stuck with me, like what's driving these patterns and how might they affect Alzheimer's biomarkers associations. Because while it was interesting we found these in a large cognitively healthy cohort, these findings are consistent with a broader literature on sleep health disparities. So this is a map showing the prevalence of short sleep in a CDC study from 2020. So short sleep here is defined as people reporting getting less than 7 hours of sleep. And over 390,000 people responded, so it's a pretty amazing data set. But they were able to look at what demographic factors are associated with short sleep. So they found lower household income, living in a less urban area, not having a college degree, and identifying as non-Hispanic Black were all all these qualities were associated with a higher likelihood of being a short sleeper. And so I think you can see where I'm going with this is that all of these qualities are things that have traditionally not been well represented in our Alzheimer's biomarker cohorts. And so I wanted to, in the theme of this FRS, understand how cohort heterogeneity, as we increase the representation of these factors, might matter for understanding the sleep and Alzheimer's relationship. So the question for the rest of my talk is going to be very simple, and it's really just how will cohort heterogeneity impact the association between sleep duration and amyloid PET? So you've heard a lot about the makeup of the pointer study. My sample is focused on not just the imaging ancillary study, but people who also volunteered for the sleep ancillary study, which, so the overlap of imaging and sleep was 426 people, which is really exciting. And these people, I'm going to be focusing on the questionnaire data today, but they also have actigraphy and sleep apnea data. And then also the A4 screening study. So we see many of the same differences that we saw comparing to the other cohorts where people in Pointer were younger. They had lower years of education. We know that representation was higher in terms of people from underrepresented groups. They had higher BMI, and centiloids were lower, so lower amyloid PET burden in Pointer relative to A4. The way that sleep was quantified was slightly different across studies. So in A4, people just responded with one number, the number of hours of sleep they usually get, whereas in Pointer, this data was available as bins. So you can see everyone who slept 7 hours or more was collected in one bin. But what's really interesting here, I think it's pretty striking that the people in, here's this little thing, the people in A4, you can see that over half of the study is getting 7 or 8 hours of sleep, whereas in Pointer, the 7 or more group is actually less than a quarter of the study. So we saw when we dichotomized this just as short sleepers and more than 7-hour sleepers, this is really a quite striking difference. And I'll remind you that Pointer, while there were many different criteria for inclusion in this study, including having a sedentary lifestyle, there was nothing about your sleep behavior that involved how you were recruited. So I'm going to show a couple pictures to highlight these differences across the cohorts. And the patterns I think are interesting. So you can see in the red that the short sleeper proportion is much higher in Pointer than in A4. You can also see that there's somewhat higher proportion of people with lower than 14 years of education, but the patterns between education and sleep are the same in both cohorts, where you see 72% of people in the high education group in Pointer are short sleepers, but it just goes up to 82 in the low education group. In A4, we see 25% high education group, and then 31% of the low education group are short sleepers. And something similar when we dichotomize the non-Hispanic white participants and participants from underrepresented groups. Again, we see a higher proportion of representation in Pointer, but again, similar patterns across cohorts, 72% being short sleeper of the non-Hispanic white in Pointer. It goes up to 83% of the underrepresented population. And in A4, this goes from 25% of the non-Hispanic white to 43% in the underrepresented population. So I think I just want to illustrate here that even though we have these proportional differences in demographics, it's certainly not explaining the difference in sleep behavior across the two cohorts. Okay, so to get to it, this is pretty similar to the plot I showed you before, showing this sleep duration and centiloid effect in A4, but I do want to highlight that we are adjusting for those same demographic factors, so education and self-identified race and ethnicity. And in Pointer, we actually do see the same effect. You can see overall, as I mentioned, the levels of amyloid are lower in Pointer, but I think it's pretty striking, smaller sample, but a similar effect. And then to look at this a different way, we did a logistic regression predicting amyloid positivity, and with sleep duration and the demographic factors in the model, and we found that sleep duration, short sleep duration and older age were both predicting amyloid positivity, and belonging to an underrepresented group was predictive of less likely to be amyloid positive, as Talia mentioned in the previous talk. And we actually see the exact same pattern in A4. So given this ethno-racial effect, we wanted to break this down and see as just one of the groups driving this effect. And we found that this is kind of an issue of sample sizes, where it seems like the groups that have the largest samples are driving the effect. You can see in Pointer, there are very few non-Hispanic Asian participants, at least in this subset. But in general, the directions are all going the same way, where people who are not non-Hispanic white are tending to have less likely to be amyloid positive. Okay, so the next thing that I wanted to do was to stratify the sleep and amyloid relationship by the different demographic factors that we know are related. So first, this is just showing, so only in the non-Hispanic white participants, we see a relationship that's very similar to what we saw in the overall cohort. But we look only in the non-Hispanic black participants, the sample size is very small, or was relatively small compared to the other plot. We do see a very different pattern, almost suggestive of a U shape. When we look in A4, we see something similar, where in the non-Hispanic white group, it's the same pattern as the overall cohort. But in the non-Hispanic black group, here we actually see the opposite effect, where there's a positive association between how much they sleep, and so people with more sleep are more likely to have higher amyloid. And this actually reminded me of a paper that Casey Dieters did on the A4 screening data, showing that there was bias in who got into A4. People with more comorbidities, were less likely to be screened. And this kind of suggests to me, are the people who are sleeping poorly and have a lot of amyloid maybe not making it into this study. And in the non-Hispanic Asian groups, we don't see quite as striking an effect, but certainly a different pattern than in the overall sample. When we look at education, it's very similar, and these years of education and ethno-racial group was not overlapping. In either of the studies, but again, we see in the high education group the same pattern as over.